And Maria Lopez is professor of microbiology at the University of Almeria. She is head of a research group working on biotechnological valorization of organic waste in the Center for Research in Mediterranean Intensive Agrosystem and Agri-Food Biotechnology. She got her PhD in 1996 in pharmacy. Her research is focused on the study of microorganisms that produce lignocellulose degrading enzyme and the use of microorganisms for bioremediation and biorefinery. She has been principal investigator of 4 national projects, 3 EU projects in Frame Program 7, LIFE, and Horizon 2020, 1 USDA project, that is the United States Department of Agriculture in USA, and currently coordinates the BBI Horizon 2020 Recovery project that was presented yesterday. She published more than 80 scientific papers, 9 book chapters, and has 3 patents. She is a member of the Senior Advisory Board of 3 scientific journals and senior member of the Spanish Society of Microbiology and the Spanish Composting Network. And she's also a great friend. We met many years ago, 2001, in Peoria, Illinois, at the USDA, where I was also staying for a stage, and she was there too. And so it's a long-lasting friendship, cooperation, even in European project and in research activity. So let me give the space and the word to Professor Maria Lopez.
Thank you, Patricia, for your presentation, and good morning, everybody. Thank you for inviting me and give me the chance to show our results. And also congratulations for this conference. I see the agenda. It is very, very interesting, the sessions. And I'm sure you are having very good time there. I wish I were there because, well, unfortunately, I could not join. I will share my presentation, if I can do it. Do you see my screen?
Yes.
Okay. So this work that I will be sharing with you are some results of the project that Giovanna presented yesterday. Is a Recover project. This work has been done by, thanks to the collaboration between the University of Almeria, the University Miguel Hernández, and the University of Pisa. And it is the power of synergistic interaction between organisms for plastic recycling and degradation. For this talk, first I will show you a little bit more about the project. I'm sure, well, yesterday Giovanna explained it to you, but just to put in context this work, the objective of the work and result that we have obtained on the use of microorganisms together in consortia, and also with the insect Galleria melonella and the earthworm Eisenia fetida. And at the end, I will show you our conclusion of this work. Recover is focused on the plastic that are used in the food packaging and the agriculture, and at the end of life. These plastic are recycled, but some of them are not recyclable through the system that are currently used, the treatment, the mechanical and chemical treatment. Mainly because they are composed by a mixture of different types of plastic or are multi-laminated, and this is why they are incinerated or landfilled. For this fraction, Recover is trying to find a solution to recover this fraction. But there is an important fraction of this plastic that are collected by the waste management system that remain together with the organic fraction, and at the end after composting will contaminate the compost and will end up in the soils. This assumes more plastic contamination to the soil together with the one that is related directly to the environment. So for this case also Recover is trying to find solution thanks to the use of organisms, a mixture, a cocktail of organisms. We focus on polyolefins and polymers that are not, in a sense, are non-recyclable, like polyethylene, polyestyrene, and also polyethylene terrestrial. This is because of the difficulty to break in nature the bonds between the carbon to carbon. But there are some evidence that there are some insects, mainly insects, but also some earthworms that are capable to digest those plastic by feeding them. And the microbiome they have in the guts plays an important role. So it is expected that the insects and the earthworms partially degrade and integrate in their tissues the plastic or mineralize it thanks to the microbiome. But also there are some microorganisms that are capable to degrade those plastic both in the guts of the organisms and free living. Thanks to the different enzymes. So they can mineralize it. But we consider that using only one microorganism by one is not a good choice, more when we are considering a mixture of plastic that can be in the waste stream that we are considering. So as I said, there is evidence in that the insects, the larvae of the insect are capable to digest plastic, but also mixture of plastic. For example, this is a previous work done by Casson and also by Brandon, in which they used polyethylene, a mixture of polyethylene and polyestyrene. And it was evident that together with the action of the larvae itself, it was the microbiota they have in their gut, the one that was responsible of the transformation as well. So it is not only insects, the one that can feed on it, but also the microorganisms that inhabit their guts. So the idea was here to consider that by combining different biocatalytic systems, both microorganisms and their enzymes together with the insects or the earthworms, we will be capable to maximize the transformation yields, and this will allow also to treat mixed plastic waste stream, and in addition convert fossil fuel plastic into biodegradable counterparts in a single step. One of the focus of the project is to produce, for example, chitin from the exoskeleton of the insects. So that is the idea of this project. So in this work, we developed a microbial consortia that was expected to be capable to degrade a mixture of plastic, not only one plastic at once. And also increase the plastic conversion efficiency by the Galleria melonella, which is the glitter wax moth. You can see the picture on the right. And Eisenia fetida, which is the earthworm that is used for vermicompost production through the incorporation of microorganisms together with the insect and the earthworm. In the case of the microbial consortia, the idea was to combine different microorganisms according to the enzymes profile. We screened a very, very large collection of bacteria and fungi using different strategies, and we tested them for different enzymes that could be related to degradation of the plastic, like lipases, glutinase, and ligninase. And according to the profile and also their capability to colonize the plastic, in films, we selected 6 bacteria and fungi and combined them in pairs or in trios to build up 8 consortia. And we used them to inoculate media in which the plastic, 4 different plastic, low density polyethylene, linear low density polyethylene, polyethylene terephthalate, or polyestyrene, were used as the sole carbon source for the growth of the microorganisms in the consortia. And we measure the growth after 7 and 14 days. Here you see the graphs of the growth level reached by the bacteria components of the consortia or the fungi, in the case the consortia has fungi as components. And you see here, after 7 days, the consortium, which is number 8, was capable to grow from all the plastic tested. We tested the 4 plastic, I said, but in 2 different forms, well, not forms, one virgin and another recycled for each case. So this consortium was capable after 7 days to grow from all the plastic considered and also maintain the growth after 14 days. But after 14 days, there was another 2 consortia that were capable to grow from a large range of plastic. In the case of the component, the fungi, only 5 consortia had fungi. And I want to highlight the consortium number 4, that was also after 14 days able to grow from all the plastic. That means that if they can grow from those plastic, they can probably mineralize it or transform it or each of the plastic considered. So considering this, we tested the best performing consortium in another step in a flask in which after 30-day incubation, we tested the plastic weight lost. And we found out that the consortium 2, which is composed by 2 fungi and 1 bacteria, was capable to reduce the weight of LLDP recycled and LLDPE virgin at around 12%, and polyethylene terephthalate at about 2% reduction of the weight. It is amazing for the case of LLDPE and LDPE that are very, very difficult to degrade, and the reduction of the weight tell us that in comparison to other works that it is a very, very, very high degradation level. And in the case of consortium 4, we also found a reduction of weight loss of LDP and LLDP, virgin and recycled in each case. Trying to find out which is the modification, if it is mineralization or if there is another changes on the plastic. We analyzed it. Well, in this case, for example, it was in University of Pisa. Analyzed the changes in the polyethylene terephthalate after treatment with consortium 2, and there was evidence that there was a chain scission through the hydrolysis of the ester bonds that was evidenced by the changes in the chemical composition in the surface area and also in the FTR spectrum with a different, you can see here in red, the recycled C2 treated with the consortium in comparison to the control in blue. There are some different bands that demonstrate that there was some excision of the hydrolysis of the ester bond that can be related to the microbial degradation of this polymer. In the case of the LLDPE, there was also, in this case, small changes in the chemical composition that were observed, some oxygen functional group that was observed in the case, mainly in the case of the plastic that was treated with carbon, with consortium 2. And in this case, in the case of LDPE, there was no changes evidenced. Those analyses were done. I forgot to mention that the plastic was used as a powder, not as a film. So seems to be changes and maybe modification, mineralization, sorry, and but only small changes were evidenced according to this analysis. Going through the use of microorganisms together with the Galleria melonella, first the approach was first to isolate the microorganisms that are singular for the larvae that are fed, as you see here in the picture, that are fed on plastic and are not present in the larvae that are not fed on plastic. And we selected microorganisms that were singular for those larvae and produced them and add them to boxes in which the moth, the larvae were feeding on plastic. First, trying to introduce those microorganisms to increase the population of plastic degrading microorganisms inside their guts. And after reading the larvae, the survival of the larvae was obtained after 14 days breeding. In previous analysis, in previous assays, it was demonstrated that this insect is capable to feed on, as you can see here in the film, to feed on film, plastic film. And the result demonstrated that the use of the bacteria together, the selected bacteria together with the larvae, were capable to grow from LDPE 100% only LDPE, millet LDPE, and reach a similar growth level or survival level as it was obtained with the standard diet not containing plastic, both for the two bacteria that were selected, as you see here in the picture with the plate petri dishes. In the case of the larvae, the approach was similar. First, we obtained microorganisms that were abundant in the guts of the earthworms, and we added those microorganisms in a media in which the Eisenia fetida was grown. This has a very low amount of plastic because this is intended to try to degrade the plastic in an environment in which there is organic matter together with the larvae, with the earthworms to produce, for example, vermicompost, clean vermicompost. And we added the microorganisms, both trying to add to the microbiome in the guts, and also working, what we say, working from the outside, trying to degrade the plastic from the outside. That was one of the consortia that I mentioned before. And we analyzed the survival. Well, in this case was the partner from the University Miguel Hernández here in Spain, and they analyzed the survival of the earthworms after 14 days in contact, both without the microorganisms added, with the microorganisms that will add more to the digestive, with microorganisms working from the outside, which is called here EXO, PMC, plastic microorganisms consortia, plastic degrader microorganisms consortia, and a mixture of the one that will be working from the inside of the earthworms and from in the outside of the earthworms. And this was, in this case, the survival was even higher than the one obtained without the microorganisms added. Then it remains to be known what happened with the plastic. It is not only the survival, not only the earthworms, but also the insects are capable to survive. We are now trying to find out what happened with this plastic in this mixture, and which is the modification. of the plastic, and if it affects, it changes during the passing through the digestive of the insect. So the conclusion that we finally obtained was that the microbial consortia selected in this work are very promising tools for the treatment of the heterogeneous mixture of plastic waste. And the combination of microorganisms and Galeria melonella and Eisenia fetida allows higher survival when feeding on plastic, which can be used for plastic biological recycling. And further analysis are required to determine the fate of plastic bioconversion by the combination insects of earthworms with microorganisms. So I would like finally to thank all member consortium, of course, the co-authors of this project and the financial support of the DBI EU and Horizon 2020. Thank you for your attention. You can see here my contact and the project website if you want to know more about it. Thank you very much. That's all from my side.
So thanks really, Maria, for this very interesting and innovative presentation. We're all very attracted by this kind of study. And now we have some time for questions. So you just see Francisco. And first,
Thank you,
Francesca.
Maria. Hello, it's Francesca
Hello.
speaking. Francesca from
Hi,
Arca.
Francesca.
Hi, good morning. So I'd like to ask you if you consider, as far as I can see, you provide as food a film, plastic film. So it is possible to provide to the insect and earthworms and so on some powder just to evaluate if they can enjoy it better than the film, or if they can eat in the same way or better. I don't know.
Well,
What do you think?
Thank you, Francesca, for your very interesting question. In fact, the picture I show with the film was a preliminary analysis. The one that the results I show you, I have just explained, is using powder instead of films. It is easier to see that insects are feeding on the plastic when you use film. But for most of the assay, we are using powdered film, powdered plastic. And now we are trying to know. In fact, they are in University of Pisa, and also Iris is analyzing, which is another partner of the project, analyzing, trying to know what happened with this plastic, where it goes, if it is mineralized or the remaining plastic, which is the changes. So we need to know, because we want also to be sure that there is no microplastic after passing through this system. So that's it.
Many thanks, Maria.
Thanks you.
Good morning, Maria. My name is Francisco Villaplana from KTH in Stockholm. So thank you very much for a very interesting presentation. My question is maybe a bit different. I was wondering, I was interested by the data of weight loss that you show, for example, for polyethylene. And we know that in plastics, we have also additives, apart from the polymeric chains. So my question is, are the microorganisms feeding on the additives first, or together, or with the polymeric chains? Have you looked into that?
Okay, that's a very, very interesting question. We have done some analysis for some of the consortia, because we are testing very, very large range of different microorganisms. And if they are feeding from additives, they will be feeding, well, the experiment we did demonstrated that they should be feeding from those additives at the beginning of the test. We used to test some incubation last for 3 months, more or less. And at the end, well, if there is according to the growth curves, it seems like a start growing from one of the components. We need to do more tests because it is a complex thing. There is some different types of additives that it can contain. But there is a change in the growth curves. So we hypothesize that they start growing from the easiest. Well, if we can say that there is something easy to metabolize here, but they may start with some of the additives, and then after maybe 1 month, they start growing from the polymer. So also testing, we design different tests trying to demonstrate this, not with analytics so far, but according to the growth and the design of the experiment. So we think they are using the polymer itself.
Thank you. Yeah, I think I agree with you. I think they would fit first in the easiest accessible, which probably is the additives. I think it would be interesting to actually, if you could, of course it's very nice to test the mixed plastic. Of course, that's what you want them to grow. But then it would be interesting to get maybe made polymer, just the polymer without the additive, and maybe a specific blends to see what is happening. But yeah, of course, I understand that this is a very, you say, it's a very complex system. Thank you very much.
Thanks to you. And thank you for your suggestion. Very, very good suggestion.
One more question from Professor Simona Di Gregorio from University of Pisa in biology.
Yes, I'm a microbiologist, so it's a more microbiological question. I was wondering if I'm not wrong, because I'm not working on biodegradation of biopolymers. I'm working on biodegradation of other recalcitrant in the environment. So I'm not an expert at all. And especially, I'm not an expert on the gut of insects or earthworms that you used. So as far as I understood, you can isolate bacteria from these guts, and then you can exploit in the design of bioprocesses dedicated to the degradation of. But which kind of bacterial genera did you isolate? It seems to be interesting that they can, exploit the function from the guts to the environment, and so to, for example, to a bio-based process in a composting processes or something similar. So they are what they are, who they are.
Who they are. Nothing very exotic for a microbiologist. That was expected. Bacillus and Pseudomonas everywhere.
So Firmicutes.
Yeah. Well, and Pseudomonas. Pseudomonas is amazing. They work not only in the goods, but because we were looking for the cultivable microorganisms, because, well, we are also in parallel working on the metagenomic analysis just to compare the microbiome and the functionality and so on. But we wanted they to be to be capable to grow and also to be produced. And there are also some restrictions also, because considering the potential further use of those organisms, and there was some criteria that were also considered for the selection of the final microorganisms that were mainly Bacillus. It is also easy to prepare formulation with Bacillus and to feed and to add in the environment, and to do more easy things with them.
Okay, thank you for your answer.
So, more question or curiosity from the attendants? Okay, so we thanks again Professor Maria Lopez for this really interesting presentation.
Thank you.